All right.
Hello.
Happy Thursday. Woohoo! Another week. So, couple things. One, I had a conversation with McGraw-Hill today about why you couldn't see the answers to your quizzes, and it was totally my fault. So, I think I have it set up now. So, I don't know if anyone's checked. The exam should have shown you an hour after it was due. It should show you what you missed and what the correct answers were. Has anyone checked that? I feel like people are checking it right now. I see some, yes. Yeah, take a quick peek at that. So hopefully I'll have that fixed moving forward. I did change the setup, say on like that first quiz, the one that said like functional organization of the endocrine system. So if at some point someone would check, she was saying she didn't think it would let you see them just because I set it up wrong from the get-go, but I think moving forward we should be okay. So my apologies on that. Any luck on the exam? Kelsey, can you see what you missed? Awesome. Okay, cool. So hopefully we're good moving forward. Sorry about that, guys. Okay, and so then you guys submitted those exams. I will be grading those this weekend. So we should have those back by Tuesday, no problem. So what you'll look for in Canvas where you submitted the Word document or the PDF, however you did it, it'll actually let me add comments in there. So once you see a grade pop up for that, you should be able to kind of scroll through the document and see what kind of things I commented on. So take a look at that and then let me know if anybody wants to go over anything, talk about it, etc. Okay, now everyone's going to be looking. I shouldn't have said anything. Now everyone's like looking at their exam, trying to see what they missed. Okay. Hold off on that. Wait till after class. But do look through there and let me know. For example, I have one question that I need to go back and look at because I can see that like 70% of you missed it, and I'm kind of like, do we have the wrong answer chosen? So I'll be going back through there as well. Okay, so then today we are on blood vessels. There were some faces of, we didn't like this one. Okay, so I have a couple things that I was thinking that I kind of noticed that students have the hardest time with. And that's kind of where I was thinking we would go over, but I'm open if there's things where people are like, you got to help me with this. So one of those is kind of how fluids move in or out of the capillaries. So that diffusion process and the forces that are at work there tends to be a little confusing. I did want to touch base on mean arterial pressure and make sure everyone is good with that. And then again, a lot of times it's some of those control mechanisms that give people a hard time. So thinking through kind of the auto-regulation versus nervous and endocrine system. So before I jump in there, is there anything else that you want me to add to the list? Otherwise, we can we can start with this, and if something comes up, we can talk about it. Anything to add to the list at this point? Okay, I'm just gonna go for it then. So hopefully, I mean, I made another video. Hopefully that's working okay for you guys. But I was trying to explain the movement of fluid across the capillaries. Thank you. And I started realizing I'm like, I don't know if they're getting this from what I'm saying. So let's start with that. And I am going to see if I can share a screen. And we are recording for one of your classmates who can't be here. So just so you guys have a heads up on that. Okay. I have a whiteboard. Armed and dangerous. Okay, so if you think about how blood is moving through these capillary beds, it's actually kind of fascinating, right? That you have so many capillaries, you don't even have enough blood to fill them all, right? And we're going to send blood around. The key thing with the capillaries is this is the vessel, right, that we're interested in, where diffusion is taking place, right? So if you want to think about oxygen moving out of the capillaries, CO2 in, glucose out, salts, right, all of those things, how they get to the tissues is across the walls of these capillaries. So let's see how I want to do this. So we know we have, I'm going to do a simplified version, kind of like the picture that's in your book, right? We know we have arteries feeding into a capillary. Oh, not green, blue. Right, and they're going to lose oxygen as they're delivering it to the tissues. And so then the blood is going to enter the venous system, right? So venules, the smallest of the venous system. Everybody okay there? Okay, so then what ends up typically confusing people is there's two main forces at work here. So we have osmotic forces and we have hydrostatic forces. Okay. So what are we thinking of with these osmotic forces? What does that mean? What's their effect?
That's the diffusion of water.
Okay, so osmosis is the diffusion of water.
Oh.
And so, yep. And so then when we look at this like force or this pressure, we're looking at how salts are pulling, salts or proteins, right? Some kind of solute is pulling water towards it. Absolutely. So it's absolutely related to osmosis. So really, then these osmotic pressures can either be, and I don't think I went quite this far in the video, these osmotic pressures can either be in the tissue, right? So thinking about little salts and proteins and stuff out here with the cells that would actually be pulling water out of the capillary, right, and towards the tissues, right? So that would be like a tissue osmotic pressure. Okay, so they draw water towards themselves. The one your book talks more about is the blood osmotic pressure or blood colloid osmotic pressure. And so what are we looking at there?
The blood drawing water from the tissue.
Yeah, so what in the blood would actually be causing that?
Sodium.
Okay, so we've got maybe some salts in here. I'll just put like a little plus for some sodium.
Or blood proteins.
Blood proteins, that's the huge one. Particularly, do you remember albumin from the blood chapter? Right, so you get these big proteins and they actually have a negative charge on them, and they draw water into the blood. So these two forces are essentially working in opposition. Tissue osmotic pressure is trying to draw fluids up into the tissue, and the blood colloid osmotic pressure, right, is pulling that water right back into the blood. Okay. So if you're just thinking about osmotic pressures, you could picture a situation, maybe someone whose liver is failing them and they're no longer producing that albumin, right? As soon as you start getting rid of some of the pull into the blood, where does the fluid go? Out into the tissue, yeah. So if tissue osmotic pressure is stronger than blood colloid osmotic pressure, you'd expect to see a lot of fluids moving out into the tissues. You'd get edema, right? Okay. So then I'm going to put my protein back in there. Okay. The other set of pressures, the hydrostatic pressures, you have, what's the best way to think about it? It's like the force of a fluid pushing across that capillary wall. So now we're not interested in, say, salts or proteins pulling the water, but now we're interested in kind of like the pressure or the weight of that fluid pushing against the capillary. So again, you actually have a tissue hydrostatic pressure. I'm going to abbreviate hydrostatic pressure, and a blood hydrostatic pressure.
So is that generated by the heart?
Yes. So blood hydrostatic pressure, sometimes you'll see it written as capillary hydrostatic pressure, but it is blood pressure. Absolutely. Right, so in the kidney, which we'll get to in a few weeks, you have a really rich blood supply, a little knot of capillaries called the glomerulus. And so there we'll talk about glomerular hydrostatic pressure. It's still this blood hydrostatic pressure or blood pressure. So that tends to confuse people because you'll see it have like 5 different names depending on where we are, right? So that's going to be the pressure, right, that's pushing out on these capillary walls and trying to squish fluid out of the capillaries. Okay. But if you picture some like edema we were just talking about, right? So as the fluid maybe starts filling that extracellular space, it's going to start pushing back, right? And it's going to be basically so heavy, so full, kind of like you filled a balloon, right? It starts filling, filling, filling. And then if you let go of the nozzle, right, it wants to come right back in. So that tissue hydrostatic pressure, as it's getting fuller and fuller, it's going to start exerting more and more force that would push fluids back into the blood. So which 2 forces are going to increase the fluids in the extracellular fluid?
Blood hydrostatic pressure
Okay,
and tissue osmotic pressure.
and tissue osmotic pressure. Good. So the blood hydrostatic pressure, the blood pressure is forcing fluids across the wall, and that tissue osmotic pressure, the salts and the proteins and things, are drawing water out as well. Okay, and then the reverse is also true. If you want to think about what sort of things are going to pull the fluids back into the bloodstream, it's the blood colloid osmotic pressure, all that albumin and salt, right? And then any tissue hydrostatic pressure, where it's forcing the fluids back in. So when they're talking about like net filtration and things like that, it's really a balance of all 4 of these factors. Sometimes people just want to talk about, say, like blood hydrostatic pressure and blood colloid osmotic pressure, but it's actually all 4. So one of the things that students typically find frustrating is what you'll see happen is more on the capillary end of things. What you'll find is a lot of filtration. So kind of this net movement out of the blood and into the tissue, right? Because that blood hydrostatic pressure and the tissue osmotic pressure are a little bit stronger on that side, okay? But then at the end of the capillary, if you think about if all this water moves out on this end, what you're left behind with, right, are a lot more proteins, right, and a lot more salts and things. And so now, towards the end of the capillary, you'll actually see things like blood colloid osmotic pressure kind of winning. Students seem to find this really frustrating, that we do this kind of filtration and then reabsorption in the capillary bed. It's like, why not just leave it in the bloodstream? What is the point of this?
It's how you get nutrients to the tissue.
Okay, so absolutely. So we're increasing nutrient distribution to the tissues. We're kind of like bathing the tissues. What else? Joe, were you gonna add something?
Yeah, I was gonna say to supply fluid to the lymphatic system.
Okay, so this is absolutely where we're heading next week, right? Is that when you have all this extracellular fluid out here, we end up, I don't even know if I can draw it, right? We have this whole nother system, that's a lymph capillary, this whole nother set of tubes, right, where this fluid can end up moving into the lymphatic system. Do we filter so we can supply the lymphatic system, or is the lymphatic system there to like deal with the mess that we've just made? You can kind of look at it either way, right? Because the lymphatic system is going to play a really important role in then running that through lymph nodes, filtering it, looking for microbes and things like that, right? But we will, that's next chapter is the lymphatic system, and we'll look at how that kind of collects a lot of this extracellular fluid. Right? But absolutely distributing nutrients, distributing hormones, even like oxygen, right? Again, none of these things can diffuse more than like a couple cells away from each other. And so you have this really rich capillary supply, and then to do even better, you just like kind of dump everything out there, right? Give it a chance.
One
Yeah.
question. So what prevents all the nutrients that you just, and all the oxygen that you just delivered from just being like, oh, sweet, we've got increased blood colloid and mesotic pressure. Now we're just going to go right back in. Like, why?
Yep, so those forces are, the osmotic pressures are really pulling on the water.
Okay.
Yep.
Okay.
Yep. Good question. Okay, anybody else, anything I can try to make a little more clear on this idea of how the capillary diffusion takes place?
I have a question. Are each of these kind of arterial capillary connections, are they near each kind of lymph node or tissue? So how would the lymph glands, like how would the lymphatic system know? Is it like
So
three at stop signs so it knows?
no, so these capillary beds are going to be spread out, right? You need capillaries next to basically like every couple cells, and you're not going to have near that distribution of the lymphatic system, though it's pretty widespread. So we'll actually get into that next week. But these kind of dead end lymph capillaries would probably find themselves around these capillary beds, but then things like lymph nodes, you tend to have them kind of near your armpits and your groin, right? The actual filters before that fluid, that lymph gets back into the body, into the torso. So they like overlay each other, but it's not like, man, every time you see a capillary, there has to be a lymphatic there. Yep.
So, real quick, the osmotic and hydrostatic, they're both pushing and pulling fluid, correct?
Yep, osmotic pressure is pulling, right, towards the solute, and the hydrostatic pressure is pushing because it's like the pressure of the fluid.
Okay.
Yep.
So tissue osmotic pressure pulls the fluid toward it. Hydrostatic tissue pressure pushes it away from the tissue.
Yes.
Okay. Okay.
Yep. So yeah, so good question. So like tissue osmotic pressure, how could I even draw that, right? It's like, wow, yellow is a great color, isn't it? Can you even see that?
Oh, yeah.
Okay, right, so that's pulling towards the salt, right? So it pull fluids out of the blood supply. But then you said what? Tissue hydrostatic pressure. Oh, so many choices. This is fun. Tissue hydrostatic pressure, right, would be the weight of the fluid out here in these tissues pushing right back. Yep. Yeah, so those are absolutely, that's a good point. They're opposite of each other. And the same is true in the blood. Blood colloid is pulling into the blood. Blood hydrostatic is pushing out of the blood. Okay. Good. Anything else there?
So nutrients and oxygen and carbon dioxide follow the water.
Yeah, there ends up kind of being this. So oxygen diffuses right across cell membranes. So this probably isn't as important, but yeah, there ends up being kind of this bulk movement of fluid, and so salts and glucose and things like that are going to go with it.
Okay.
Yep.
And then on the venous side, or when it switches, when it's sucking water into the vein, then it's pulling like waste.
Yeah.
And
Oh, that's a good point too. Yeah.
that's going in with the water as well.
What's that?
That goes with the water,
Yeah.
that process. Okay.
Yep. So there's some of that. Good. Okay. I was very excited that I figured out how to use my whiteboard. So you'll be seeing a lot of the whiteboard now. My poor micro students, they've already dealt with this enthusiasm this morning because I was like, yes, I got my whiteboard. Okay. Oh, who was next on my list? Oh, let's look at this real quick. Maybe. Right, I'm skipping all the anatomy. You can read about that again if you don't remember the difference between, you know, who's got valves and this kind of stuff. But I did want to, whoop. I did want to touch base and make sure, because I know some of this got a little bit mathy, right? So this idea that blood flow is proportional to the change in pressure over resistance. Can someone put that into their own words? Like, what does that mean for us? And maybe give me a hand or a thumbs up if you want to answer that. Who can put that into their own words, what this equation means? Molly, what you got?
I don't know if this is right or not,
Go for it.
but I mean, if there's a change in pressure, like if there's still a lot of resistance, it's not going to necessarily help the flow.
Okay, good.
Right? Like it'll hurt the flow. But if there's not a lot of resistance and there's not enough pressure, it's still going to not be very good for the flow either.
Good. No, those are both really good interpretations there, right? So this change in pressure, remember, so this is key, right? Blood is always moving from an area of high pressure to lower pressure, right? Even if you look at, oh, I can't see because I made you guys all big. Right, if you look at the blood pressure on this graph, right, notice, oh, you're still in my way, over there. Notice at the vena cava, this is extremely low blood pressure, and it still flows into the right atrium. And so that means that that right atrium has even less pressure, right? It's emptied itself, and it's just, right, it's just waiting for blood to flow in. So this idea that blood is always moving from higher pressure to lower pressure, right? This graph also explains that blood moves out of the elastic arteries and into those muscular arteries because they have lower pressure, and from the muscular arteries into the arterioles because there's lower pressure, right? And into the capillaries because there's lower pressure, right? So when you think about your equation, this idea of the change in pressure, yeah, I think it's going from higher pressure to lower pressure, but say, you know, this elastic artery, like the aorta, right? If you want blood to actually flow through the aorta, you need to have that drop in pressure, but it needs to be a big enough drop. This is where Molly was going, to overcome that resistance. Okay? So someone with hypertension, right, if all those muscular arteries are just like clamped down, right, making a really small radius, that's upping your pressure. Blood doesn't want to flow. It's hard to flow against that resistance. And so the only way to get it to flow is to make sure your high pressure is high enough, right, that the difference can overcome that, which means your heart is working really hard, right? Questions on that? Yeah, go ahead, Courtney. Say again, Courtney.
It's like the water slides at like hydraulic energy, you know, you have to get the slide high enough to get the water up it to go over the other hill.
Yeah.
Kind of.
Yeah, absolutely. Yep. So because that hill is then your resistance.
Right.
And so you have to have enough speed, you have to have enough kind of change in pressure from that first hill to make it up and over the next. Yeah. Yep. And so really when you think about this, and I can't emphasize enough that blood flow is the important thing here, right? You need the blood to be getting to these capillary beds in all your organs, right? You want that blood in the liver and in the brain and those sorts of things. And so then really you only have two choices. You either figure out a way to decrease this resistance, right? Or you have to increase that higher pressure, right? Because frankly, at the end, right, you can't go any lower than this low pressure. So you either have to increase the work of the heart or figure out ways to have less resistance. And so in a healthy individual, you would expect to see, you know, what we would then call like an average level of resistance, right? There's still friction, there's still these forces, but it would be the kind of resistance that the heart's totally capable of overcoming.
Aside from just like having young, healthy veins and arteries, how else can you decrease resistance?
So what are the three factors that we talked about for resistance?
Oh yeah, that's right.
Ooh, yeah, no, what are they? That's a good.
Okay, so laminar versus turbulent flow,
Okay,
turbulence, viscosity, and diameter of blood vessel, and length of vessel.
So this idea of laminar, it's not going to let me fix that now. Laminar flow versus turbulent flow, right? Laminar flow gives you less resistance, so that's ideal. So what do we do to avoid turbulent flow? What causes turbulence?
Stress,
Why would stress cause turbulence?
because it starts just pumping your blood faster.
Vasoconstriction.
Yeah.
So stress could change things like the heart rate, right, and blood pressure, but the other place probably that stress would come in is on that vessel diameter, right? So sympathetic nervous system, right, the SNS, oh boy, I shouldn't be trying to, the SNS is going to cause vasoconstriction. It's going to decrease the diameter, and that increases the resistance, right? It would be harder to like blow out through a little coffee stirrer than it is a regular straw. So absolutely, stress could stimulate the sympathetic nervous system. That would decrease diameter, but it's probably not going to increase our turbulence. Where does turbulence come in?
I think you mentioned that smoking causes turbulence. I wasn't sure how, but I think you mentioned that.
Okay, yeah, so any damage to the endothelium, right? So blood should be, do we have that? Come on. Right, you want blood, oh wow, I got all sorts of stuff on here. You want blood to be flowing nice and smoothly through this tube, and anytime you hit any kind of damage on the endothelium, it's gonna kind of throw it into those pools and eddies. And so some of this is totally natural, right? Like around the valves in the heart, there's always turbulence, right? There's just no way around it, but you want those vessels to be nice and smooth and clean. And so some of the chemicals in smoking cause endothelial damage, might just be a little thing. But then if it starts to turn into a plaque, right? If platelets maybe come to that site of damage and you start building up like a thrombus, a clot on the side there, that would cause turbulence. Atherosclerosis, when you start building these actual like fatty deposits on the sides, those are going to be both narrowing the vessel, right, which increases resistance, and causing this turbulence. So that's kind of a double whammy. Okay. So we want less turbulence, so don't smoke, right? Keep those arteries nice and clean. Decrease your stress, right? Keep those blood vessels relaxed and open and able to allow blood to flow through. What was our other one?
Viscosity.
Oh, viscosity. Yeah, viscosity. So do we want high viscosity blood or low viscosity blood?
Low.
Okay, what makes the blood, how would we measure the blood's viscosity?
Hematocrit.
Hematocrit, good. right? So hematocrit is that percentage of the blood that's red blood cells. So if you're at 45%, you are less viscous than if 60% of your blood is red blood cells, right? That's starting to get pretty thick and sludgy. So I would say there's like a sweet spot, right? You don't want like super low, because that would mean you have a really low hematocrit and then you're not carrying oxygen, right? You don't have the cells for it. But absolutely, if you increase those red blood cells, you are increasing the viscosity and you're going to make it harder to pump blood.
I have a question.
Mhm.
So, like being at high altitude, it's better to have a higher hematocrit, right?
Yep.
So, why are you encouraged to increase water intake at high altitudes to stave off altitude sickness? Like, how does that?
Ooh. Yep. So when you first come to altitude, what your body first senses is what?
Low oxygen.
It's the lack of oxygen. And so you increase your respiratory rate to make up for that. And actually your lung surfaces are a huge way that we lose water vapor. And so it's easy to become dehydrated as your body has increased that respiratory rate. So that's probably a lot of the encouragement to drink fluids, right? The red blood cells, the increase in red blood cells, that's going to take a couple weeks until, remember, endocrine system is more of that long drawn out gradual effect. And so EPO is going to have that effect over a few weeks. So most of it's just a dehydration thing. Yeah. So yeah, with viscosity, and this would be a problem with blood doping, right? If you allowed your red blood cell numbers to increase dramatically, your hematocrit to come up, you're super excited because you can carry all that oxygen. But what happens when you increase the viscosity? What does that do to our little equation?
Higher resistance.
Yeah. Whoop. And again, if you have trouble picturing how this works, right? Like, oh, I'm not even gonna try. That's gonna be a bad example. I'm always in like, okay, the numerator and the denominator, and how does this work? Right. Make up fake numbers if you have to think through this, right? Okay. Okay. Other questions on what's allowing blood to flow? Ooh, out of turbulence, viscosity, and diameter, which is the strongest factor?
Diameter.
Diameter.
Diameter. Good. So it turns out that those changes, vasoconstriction and vasodilation, are hugely important, and they don't have to be dramatic changes in order to make huge changes in that resistance and therefore blood flow. Okay. What's next then? Ooh, mean arterial pressure. Okay, I am really hating the way that your book described mean arterial pressure. I think we talked about that last week, right? So definitely, so I pulled this picture from my previous book, right? Notice that mean arterial pressure is the red line here, and it's showing you where that falls kind of between systolic and diastolic blood pressure. And it's definitely closer to diastolic, okay? So mean arterial pressure, did I give you the actual equation for this?
Yes.
I'll give it to you again. Mean arterial pressure, right? You've got diastolic pressure plus, this is going to be hard to do on here, pulse pressure, which is systolic minus diastolic. Right, systolic minus diastolic is this pulse pressure that they're pointing out here, right? So the difference between what kind of pressure, say, the aorta is feeling when the heart contracts versus when it relaxes. And this is all over, divided by 3. Let me try drawing that up too, and see if that makes more sense. So mean arterial pressure is diastolic blood pressure plus systolic minus diastolic over 3. So let's just do this. Let's do one together. again, like you're not, as a nurse, you're not gonna have to sit there and calculate this. This is gonna show up on a monitor, but let's take a peek at what this might look like. So what's normal blood pressure?
120/80.
120/80, very nice. And so which of these values is systolic and which is diastolic?
Diastolic is
120 is systolic
No,
and
yeah,
80
there
is
we go.
diastolic.
Okay, so 120 is our systolic, right? The higher pressure, because remember, systole is when the heart is contracting, right? So how much pressure can it exert? And then diastole is when it's relaxed. So that's gonna be your lower, your bottom number. Okay, so let's try to calculate this. Anybody have a calculator with them? Maybe on your computer, maybe on your phone? Okay, so setting this up, right, I would say, okay, I need to know diastolic, which is 80, plus 120
It's 120.
minus 80. But then all of that divided by 3. So what is 120 minus 80? Don't need a calculator for that one. 120 minus 80. 40. Good. That's okay. So we have to do 40. This is where the calculator comes in, right? We need 40 divided by 3. And then we're going to add 80 to it.
I don't have a calculator.
It's 93.333.
That's what I got too. So what? 40 divided by 3 would be around 12, 13, right?
That's where you get the 13.3 repeating.
Yeah. So there's the 93, right? So 93 would be our mean arterial pressure. That would be a really average number. If you fall below, I've seen different numbers, 60 or 65. 65 is probably the one I've seen most often. So you need a mean arterial pressure of, well, is that greater than or less than? Greater than, someone help me, greater than or equal to 65? Did I draw my alligator the correct way?
Yeah, you did.
Thank you, Kelsey. Right? You need a mean arterial pressure greater than 65 to actually have enough then, right, decrease to keep blood flowing, right? So if pressure is too low, flow stops. It can't overcome resistance. So 65 is kind of right around your magic number there. If you're below that, what's happening to those organs?
They're not getting enough blood.
They're not getting enough blood, so they're not getting enough oxygen, nutrients. What happens to, say, your liver?
Eventually necrosis.
Yeah, it's good. The cells are going to start dying, so you start going into organ failure. So this is the problem with something like shock. Someone's blood pressure is too low. Their mean arterial pressure has dropped so low that they're not able to actually perfuse their organs. So let's try this. I'm going to put you in breakout rooms, and you have a blood pressure now, a patient with a blood pressure of 80/40. Everybody have this equation? Hopefully someone in your group will have written it down. 80/40 is going to be your new blood pressure. I have to stop sharing so I can put you in groups. Work with your group and see what the mean arterial pressure is. Jennifer, oh, there you go.
Does
Um,
anybody in the group need to be like walked through it at all? Or do you go, all got it?
I feel good about it. I'm not sure how, who's our third here? Wait, we got 5?
80 minus 40 over 3.
Okay, there we go. I was doing it separately. Thank you. I was like,
Yeah, I did it backwards the first time too.
okay, this is a lot better. Thank you.
Okay.
What'd you guys get?
53.3.
That's sounding like a problem, isn't it?
It sure is.
Okay. I'm going to get everybody back to the main room. Looks like breakout room 1 is still working. We'll give them a second here. There they are. All right. Anybody think your group got it? Anybody want to share? Okay. Yep. I'm getting some thumbs up. Jennifer, what'd you guys come up with?
53.333.
All right. Everybody, does that sound right? Is that what other folks got? Okay. Cool.
Okay.
Good deal. And is that a good number? I'm saying no, right? So that person is not getting perfusion in those organs. So again, right, it's actually fairly simple math once you've done the equation. I doubt that you're going to be asked really to calculate that. And honestly, you probably start to get familiar enough with blood pressures that you could see someone's blood pressure and be like, those are bad numbers, right? Without even doing the math. So, but I did want to make sure you guys were comfortable calculating that. I might ask you to calculate that, just to make sure. Okay. All right. Any questions about mean arterial pressure, what it means? Okay. Doing good. All right. So I think, let me see where I am. So many pictures. Okay. How do I get back to you? Okay. So then the last thing I really wanted to touch base on is this idea of, again, the regulation of these responses. And there's, hopefully you guys all noticed the overlap, right? They're super tightly tied with what the heart is doing as well. So again, a quick reminder that we'll see this over and over again. Every organ is going to try to fix the problem itself, and that's autoregulation, right? And then if that's not enough, we're going to call in the nervous system and the endocrine system. And your book was using terminology more like short-term regulation versus long-term regulation, right? Of the nervous and endocrine, who's short-term?
Nervous.
Nervous.
Yeah, good. Yep, that's the nervous system. Okay, so you can think of those two kind of interchangeably. Okay, so how are we feeling? Well, no, I'm going to phrase it differently because you're all just going to sit there and look at me. What are some examples of autoregulation? Whoop, sorry.
Vasodilation of precapillary sphincters, right?
Okay.
Yeah.
So you could have vasodilation of those precapillary sphincters really at any of these levels. What sort of things is it that's going to cause that vasodilation that's auto-regulation, that's the kind of the capillary bed itself taking care of itself?
Vasoconstriction and the other one?
Vasoconstriction and vasodilation, what's triggering those? What chemicals are we responding to?
Like,
Zoe.
I get confused. Oops,
An increase in like CO2 or decrease in oxygen.
Good. So, and notice what that means, right? The capillary bed is sensing when oxygen is getting used up by working tissue, right? Or when that tissue is starting to dump CO2 in. And so either of those chemical signals would tell that precapillary sphincter to vasodilate. We want to deliver more blood to that specific area. Isn't that cool? Like such a localized little adjustment that your body can make, right? And it kind of makes perfect sense. There's a couple other ones, things like lactic acid. Again, that would be a signal that the cells are doing fermentation, right? They're doing an anaerobic respiration. They don't have enough oxygen. Okay, so we're going to try to deliver more. Things like increased temperature, right? Okay, and then if that doesn't work, right, if that's not enough, we still don't have enough oxygen going to those tissues, then we call in the big dogs, right? We get the nervous system, we get the endocrine system involved here. So same region of the brain is running this as was running the heart, right? What region of the brain is that?
Medulla oblongata.
The medulla oblongata, right? So that's a really good thing to keep in mind, that this not only has those cardio-regulatory centers, right? The ones that we saw adjusting heart rate, stroke volume, things like that, but also what they call the vasomotor centers. What does vasomotor mean? Zoe?
Does it control the vasoconstriction or dilation?
Yeah, absolutely. So the motor, think the muscle, right? The muscle, the smooth muscle that's there, and then in the blood vessels. So the smooth muscle in the blood vessels is what's being controlled by that vasomotor center. So telling it whether to dilate or constrict. Good. Okay. So one of the things that gets, I would say, like the most confusing in here then is that all of this is happening at the same time, right? So, and we don't even know necessarily what's changing inside of us, right? So the cardio-regulatory center is hearing, I don't have enough oxygen. And so we might be doing things like increasing heart rate or increasing stroke volume, right? Trying to increase cardiac output. And then the blood vessel, right? The blood vessels are being told who needs to open up and let more blood flow through. So you put the mesh of those together, and there's actually a lot going on, a lot of different ways, permutations that the body could take of those factors to make sure that we're delivering blood where it needs to be. Okay. So as far as the nervous system goes then, typically, right, since we're in the autonomic nervous system, we look at the sympathetic and the parasympathetic nervous system. What are those two branches of the autonomic nervous system do to the blood vessel diameters?
The parasympathetic relaxes them, and then the sympathetic contracts them.
Good. Yep, so sympathetic is going to be causing vasoconstriction. Parasympathetic is going to be causing vasodilation. Good. One thing that tends to confuse people at that point is you go, well, wait a second, I thought if I constricted a vessel, it was going to be harder to get blood flow. Keep in mind, this isn't the same throughout the body, right? And so we might be doing things, say, with sympathetic nervous system. Oh, how to explain this, right? You think about epi and norepi. What cell you attach to, what receptor you attach to might actually cause a different effect, right? So when the sympathetic nervous system is activated, we're trying to send blood to, say, to like skeletal muscle, right? To fight or flee. but we're going to shut off the blood supply to things like the digestive and the urinary tract, right? So it's a little hard, right, when you read something like, oh, it causes vasoconstriction. Well, it's not like everywhere is acting the exact same. Okay, same thing then, parasympathetic nervous system, that vasodilation, we're really thinking about that taking place in places like the digestive tract and the urinary tract, right, and really increasing blood flow to those regions, while at the same time we might be shutting off some of that blood to the skeletal muscle because we're in a relaxed stage. So that gets a little tricky. The other thing to keep in mind, or one of the things I tend to think about, is what you might see someone, if someone, say, was in anaphylactic shock, right? They got stung by a bee, they had massive vasodilation throughout their body, right? They're having this allergic reaction. And now instead of like their eyes and their nose running, all of their blood vessels have kind of opened up and fluids leaking out everywhere, right? So their blood pressure is dropping. We are not going to have good mean arterial pressure, right? We're going into shock. What does a paramedic do for them?
Gives them epi.
Gives them epi, right? And so what you're trying to get, it kind of mimics what that sympathetic nervous system response is, or what the adrenal medulla would release to cause vasoconstriction. And so you're trying to fix this, like everything is just kind of sagging out there, and bring it back in so that the amount of fluid that we have actually can exert enough pressure to perfuse organs. So sometimes thinking about how epinephrine causes that vasoconstriction could be useful in remembering that sympathetic nervous system is vasoconstriction. Okay. But that's short term, right? And again, you're making all these little permutations all the time. I think, to me, the most fascinating part of this, and maybe the most complicated part, is really what the endocrine system does. So let's get a little list going here. So if we think about the endocrine system, oh, okay. Endocrine regulation for vessels. What sort of hormones are we talking about? Let's just make a list and then we'll see if there's any questions on how they work. Who, give me a hand or a thumbs up if you have a hormone that you'd like to share with us. Carolina?
I was going to say ADH.
You got it. I saw a hand, so you get it. Antidiuretic hormone? What's it? Molly?
Was it the something that came from the atrium? From the right atrium?
Yes, atrial natriuretic peptides. Did I spell that right? So atrial natriuretic peptide or brain natriuretic peptides, you'll see those abbreviated A&P and BNP a lot. Okay.
Renin.
Okay. Zoe, did you have one? I couldn't tell if you had a hand or not.
No, I was going to say renin.
Okay, cool. Thinking one more. Joe.
Angiotensin 1.
Okay, so angiotensin 1 is going to be in this whole renin thing. So we'll talk about that. And then Taylor, did you have one?
Aldosterone.
Aldosterone, nice. Oh, you guys have more than I do. How about Kelsey in your high altitude?
What'd you say?
Zoe?
The EPO.
EPO, good. Erythropoietin?
Yes.
So isn't this fun? We're like only a few chapters out of the endocrine system and we're already reviewing it. You guys are like, sweet, I know some of this. Okay, hopefully we know some of this. So the ones that we talked a lot about in the endocrine system are maybe more straightforward, right? So what do we know, say, about ADH? How's that going to change this cardiovascular system? What's going on in our blood vessels? Who's got this one? Molly?
It's going to increase pressure because we're retaining water, helping us keep water in our system.
Good. So we're increasing water reabsorption. And let me make this bigger. Kidneys. And good. So anytime you have increase, so that's going to increase blood volume, which therefore increases blood pressure. Okay, good. What's another one we talked a lot about or talked about some in the endocrine system? Aldosterone, maybe? What do we know aldosterone does? Joe?
It increases sodium retention in the kidney, which also then pulls in water.
Good. Yep. So if the kidney is holding on to sodium, water follows sodium. And so really, right, ADH and aldosterone are often working hand in hand. And so if you really want to increase your blood volume and blood pressure, you release both of them. Increase reabsorption of sodium and also of water. Boom, golden. Okay. Zoe, Kelsey, my EPO high altitude folks, what do we got there?
It increases the generating of red blood cells.
There you go.
Creation of red blood cells.
Yeah, we increase red blood cell production. So therefore we can carry more oxygen, right? We're trying to fix that problem. So we got the good. Okay. Okay, so we have atrial natriuretic peptides and then renin. I can't remember how much we talked about renin with endocrine system. No? Probably, okay, probably not. Because we probably said something like, oh, it's like a secondary role of the kidney or something like that, is to release this renin. Okay, so this is probably the most important one, renin. I've been told by people in nursing school that like all you talk about is RAS. So let's run through it. So, oh, I want to draw now. Oh. RAS. The renin-angiotensin-aldosterone system. This is going to be your friend for apparently for much time to come here, right? So RAS is the renin-angiotensin-aldosterone. I feel like there should be commas or something, but it's all out there. Aldosterone system. Okay, so this is a sequential series of things that are going to happen that the kidney starts off, right? So I can draw a kidney. Totally. Boom. So the kidney is who is releasing the hormone renin. Renin, same thing. In response to low oxygen levels, right? So this is another one where the kidney's like, I don't think I'm really getting a good enough blood supply here, right? So in response to those low blood oxygen levels, the kidney is going to start this whole series of events. So this hormone, renin, starts circulating in the blood, and out in the blood, it is going to come in contact with a protein called angiotensinogen. I don't like how I'm writing this. Hold on. Let me think about how do I do this. Okay, hold on. Okay. Sorry. So the liver, which looks like a sideways kidney, apparently, the liver is constantly producing a protein called angiotensinogen. Yeah. right? And that ending tells us what?
Precursor molecule?
So it's a precursor. It's an inactive protein. It needs to be activated. And so and I'm trying to think of like how I want to draw this. You might come up with your own plan here. But what activates angiotensinogen is renin, right? And so we are going to convert it. Oh, this is how I want to do it. We are going to convert angiotensinogen into angiotensin 1. Right? So typically sorry, I'm making a mess of this. Typically the way I like to draw things is like an arrow is indicating a reaction. And so I'm going to put renin and my kidney on that line, right? To show that it's actually the renin being released from the kidney that's converting angiotensinogen, which is circulating all the time, into angiotensin 1. Sorry, I should have thought that through before I started drawing.
Wait, sorry, which one is circulating all the time?
The angiotensinogen. So that's just a normal liver product, and it's an inactive protein. So you can just keep it hanging out in the bloodstream. And I've already taken up my whole whiteboard here. Okay. So I'm going to write over here. Keep it linear. Angiotensin 1. If you have room, you can just keep going, but I'm out of room. Angiotensin 1 is going to get converted into angiotensin 2. By a hormone mainly found in the lung. Does anyone remember what that hormone is? Or sorry, enzyme. It's converted by an enzyme in the lung.
Is it just angiotensin converting enzyme?
It is angiotensin converting enzyme, abbreviated ACE, angiotensin converting enzyme. Does anyone know how this relates to the current world pandemic?
Can I assume that COVID affects this?
Yes. So this enzyme is embedded. Yes, you can. So this enzyme is in the lung tissue. It's sitting on the outside of cells, waiting for angiotensin 1 to come by. And it's the same protein that COVID uses, that the SARS-CoV-2 virus uses to dock and gain entry into the lung cells.
Oh.
So one of the really fascinating things that started happening is that people who, oh goodness, I'm going to go way too far. People who have high blood pressure, they might be on drugs called ACE inhibitors, drugs that actually block this enzyme. And so there's been some like, at first people were like, oh crap, I think all these people with high blood pressure are screwed. Like maybe this giving them this drug is actually bad, but they've been doing studies and we think that those ACE inhibitors are actually protective, right? So helping prevent worse outcomes from COVID. So anyway, I haven't seen anyone who's using it as like a frontline drug, but they're definitely telling people who are on it to keep taking it, that it seems to be beneficial. So anyway, sorry. By the way, I hate COVID the rest of as much as the rest of you. I just really enjoy kind of nerding out on the weird stuff like that. Okay. Angiotensin 2 is going to do really two main things. I'll give us two arrows here. Angiotensin 2 in itself is a potent vasoconstrictor. So it causes vasoconstriction. So the kidney was saying, I'm not getting a good enough blood supply. And now we are gonna like tighten up those blood vessels and try to get the blood heading towards the kidney, okay? It also triggers the release of aldosterone. right? So it's not getting converted here, but it is triggering the release of aldosterone from the adrenal cortex. And then aldosterone is telling us to retain sodium, right? So the renin angiotensin, and this A is really like angiotensinogen, angiotensin 1, angiotensin 2, right? Renin angiotensin aldosterone system. Okay, so this is a major player in regulating blood pressure. And again, don't feel like this is necessarily like this catastrophic event. You are always making these like little adjustments to how much renin you are releasing in order to make slight adjustments to like vasoconstriction and how much aldosterone, how much sodium is being reabsorbed, right? So this is a really long-term kind of thing, where you'll run into some, you guys will see this so many times. One of the things we get into in patho, we start talking about things like heart failure, and you end up in this like cycling of RAS. So it just keeps trying to up and up and up blood pressure, which isn't healthy, right? If the heart is already struggling to pump blood, we don't need to give it higher and higher pressures to work against. So this will be your friend. Come up with it. Hopefully your notes look better than mine there. Did they give us? So.
Can I ask one question?
You bet.
I'm still a little bit, the thing you brought up earlier that you said often confuses students about why does constriction lead to more oxygen perfusion?
Yep. Yep, good question. So I'll see if I can make it make more sense. So you need to have adequate blood pressure to get your change in pressure to get blood to flow, right? And so maybe even one way to think about it is that vasoconstriction helps us kind of up that initial.
Okay, that makes sense.
Okay.
Like the change in pressure divides more because this one goes up more because when you constrict the vessels, it increases the blood pressure.
Yep.
Okay.
Yep.
So that makes sense.
Yep.
And I also understand that, the thing you said about shunting away from certain tissues and other ones dilate. So it's a little more complex. But
and a lot of that comes down to receptor types. And so like, I don't know if like in A&P I, like in the nervous system, if you got into things like adrenergic receptors, muscarinic, nicotinic, all those. So which tissues have which receptors can actually act really differently to some of these.
Gotcha.
Yep. In fact, think about epinephrine. Maybe this is the classic example, right? So if epinephrine causes vasoconstriction, right? It's telling that smooth muscle in the blood vessels to constrict, but then it causes bronchodilation, right? It actually tells the smooth muscles in the bronchioles to relax, and it's because they have two different receptor types.
Okay, that makes some sense, although if you constrict, then you increase your resistance.
Yep.
And so, okay,
Good.
I'll just accept it for now, but
yeah,
it's a little confusing.
Yeah, the other way I try to think about it, I hear you, is thinking about maybe even like water coming out the end of a hose, right? So like you turn your faucet on and water is just like falling out the end of the hose. But if you really wanted to direct it somewhere, if you can clamp down on the diameter, right, you put your thumb over it so it has a narrower opening, then you can increase the pressure with which it's moving. So it ends up being this weird combination of both of those. You're totally right. It's increasing resistance, but it's also able to increase that blood pressure in a way that can get it moving, get it flowing.
Okay. All right. Thanks.
No, that is a weird one.
It kind of increases the direction. Like when you vasoconstrict, it seems like the blood knows like where to go because it's it's being directed.
Yep. And so again, the way to think about it is you're opening up probably some areas and you're closing others. And so you might find that like those precapillary sphincters are open to let that blood flow in and decrease the resistance. But in some of those muscular arteries, you're decreasing the diameter to try to increase that pressure. Molly?
Right, like just because it's vasoconstricted doesn't mean it's like preventing all blood flow. Like, I mean, that might eventually happen when like you get really cold and it's trying to keep everything from going to the surface and losing body heat, right? But it's not like it's just preventing any flow at all. It's just lessening the flow. Is that correct?
Yeah, but so then that's an example that you just gave, right? Where vasoconstriction, we're actually using it to decrease the flow, right, to the surface of the skin. But then vasoconstriction in some other areas might be actually helping us up blood pressure to the point where we can propel it all the way to the kidney or the liver or something like that. And so it's this weird, yeah.
Our body's amazing.
Our body is amazing. I try not to say that except for in the nervous system, because the nervous system is so like, that I just keep being like, this is crazy that this is how your brain works. So try to have more concrete answers on the rest of it, but yep. All right. Those were like the big things where I'm like, this is where I tend to see students struggle. But you guys read the chapter, watched videos, went through dynamic study modules. Anything I can help clarify or?
So, Becky, I had to stop my video for a little bit, so I might have missed this, but I'm just curious, like, how much rote memorization and what method you recommend for memorizing all of like the veins, the arteries, their pathways.
Yeah. Oh no, that's a great question. So in my previous textbook, they actually separated the chapter that was like, these are all the arteries and veins.
That makes way more sense. Way more sense.
And so what I always did previously is I never lectured on those. That's lab. Oh, I will post the list of vessels that you actually need to know for lab, because that would be super helpful if you look through them before you come. And I just want something to write on.
Because there were a lot of, not a lot, but there was,
Yep. Yep, I will put that up right after class, the blood vessel lab sheet. So then the rest of them, don't even worry about it. And I won't ask you any of those on, say, like a lecture exam, but I thought the dynamic study module was actually decent at running through those, so I made you do it. Yeah, good question. And I would say, Allie, when you, or all of you, when you get to looking at those blood vessels, a lot of them, hopefully, if you got your body terminology down in like, like the first or second lab of A&P I is typically like, this is the brachial region, this is the axillary region, this is radial. If you have that down, then it's actually pretty straightforward because they're all mostly named after that region. So yeah, if you have that, it hopefully won't be as bad as it looks. And someone's in the chat. Cool.
That was just it's about the Bolton Strid, where a really wide river comes down to like a very deep but very narrow part, and it's like the most dangerous stretch river in the world.
Because the velocity picks up in there, and yeah. Okay, cool. Thanks, Jeff. Other questions? Otherwise, you can just go start learning those blood vessels, because there are. Yep. I still haven't heard back about the potential to open the lab up on Friday. I will let you know as soon as I hear if that paperwork's getting through. The heart model is up in the library, but I guess the library is probably mostly open during the school day. So, Molly.
Were you trying to do it for this Friday or next Friday?
The 2nd, so not tomorrow, but the 2nd. So hopefully I'll know something by Tuesday, would be somewhat useful in planning. So maybe just in general, right? Like, so if I said like morning, afternoon, evening, like who would be like morning would probably be a great time for me. That would be my best. Who's more of like an afternoon would be awesome kind of person. I see a thumbs up from, okay, 1, 2, 3, 4, 5. Okay, you're rolling in. And then who would be like, yeah, the only time I can come is like in evening sort of thing. Oh, and now I'm like, oh, that's even better. So maybe a couple folks there. So I'm really, anybody who's like, I can only come in the morning. Okay, I'll still end up sending out like a doodle poll or something to make sure we make that as open as possible. Okay, so okay, so we might be able to spread you guys out. We still can only have 9 people at a time in there, so we'll have to have a couple different openings. So again, trying to do that, but otherwise keep studying. That's coming up. Okay. Cool. So feel free to head out if you're good to go. If you still have questions, feel free to hang around. Zoe.
Yeah, I was curious because I noticed the blood lab. What review sheets are we supposed to do for that?
So the blood lab for the...
Sorry, the blood vessel lab.
Oh, blood vessel. I'll post that with the...
Because, well, you posted something saying exercise 30, 31, and 32. So, but you said review sheet optional afterwards, so I was just confused.
Okay, let me open up Canvas and take a peek.
Yeah, I wasn't sure if you wanted us just to do like the questions are optional or the diagram.
Let me take a look. I seriously, I was holding it together, and then the other day I told my patho class the instructions for their exam that were really meant for my biology class, and it's like, we're getting to that point, I think. How do I get you out of my way? Okay, under modules. Blood vessels. Okay, so we're studying circulation. Okay, so please complete the review for exercise 30 and 31. Exercise 32 is optional.
Okay,
so
I must have just kind of skimmed through it.
That's okay. Nope, you're probably not the only one with that question. So you've got those 2 review sheets to do. The third one is just if it's useful practice for you, go for it.
Okay, cool.
Okay. Okay. Yeah, Jennifer.
Just because I didn't do A&P I in lab, what else would you recommend that would be useful for studying for the practical?
Yeah.
Besides, going to the library, or if you're able to bring us in.
Yep.
Videos, I mean.
Yeah, so it depends, right? So there's definitely, if you're like, I need to see the cat again where the endocrine organs are, I guarantee if you Google, cat dissection endocrine system, there will be some professor somewhere walking you through and showing those to you. So that would be a great idea. Same thing, I can almost guarantee you can find someone walking you through a dissected heart, right? So if you're more visual, that could be great. For me, one of the things that I find super helpful is I feel like if you know where it is, then it shouldn't be that hard, right? So if you drew, wow, yeah, this is good. A cat on a sheet of paper, right? And I could say, oh, I know in the neck, the only endocrine organ up there, that's the thyroid gland, right? And then I go, oh yeah, and I know that the thymus sits right on top of the heart, right? And so if you can actually go through and sketch, maybe the cat is a terrible example, but the heart, right? You should be able to. It doesn't have to be pretty, right? It can be a box, but you should automatically know that into the right atrium, you have a superior and inferior vena cava. Or like on our cows, we really only saw superior, right? And I should know that this valve between, is everything backwards to you guys? whatever, it's the heart, it's always backwards. So on the right atrium, right, the right atrioventricular valve or the tricuspid. And so if you can get to the point yourself where you could actually, like, for some people that's super useful, draw it out and be like, totally, right? Or as you start doing blood vessels, right, draw your little cat and you're like, I know it goes from subclavian to axillary to brachial to radial, right? And so for some people, drawing it is super useful. Because I know it seems like there's a lot, but there's not going to be that many things that I could possibly, mark on the arm of the cat. So I think that helps.
Are the does the practical gonna have the vessels in it too?
Yes.
Okay, that's what I thought, but I wasn't sure.
Yep. So you get those this week, and then they're on your practical next week.
So, but just, this is probably obvious, but you're only going to be asking about you're not going to be asking about the vessels on the cat.
There will be some of the vessels that I will ask on the cat. So that'll be indicated on the list as well. So you'll be dissecting those out this week. And some of them are super obvious. You're going to be like, aha, the aorta, right? Or the abdominal aorta.
Okay, gotcha. We just haven't done that yet. Okay.
Yep. And then we have models as well.
Okay, perfect.
And did everyone notice on like some of the heart models, there were still like stickers with numbers on them? That's how the lab practical gets labeled, right? So you'll like walk in and there's going to be a piece of the heart model and you might find 3 or 4 stickers with numbers and you're like, oh, coronary sinus, right? And you go ahead and write your answer down next to that number. So I'll make sure this week that I bring what the answer sheets look like, because it really is just a sheet with a bunch of numbers and lines, and next to number 20 you write coronary sinus, and then when you see question 25 is the brachial artery, you write brachial artery. But I'll bring those sheets so you can get a look at that. I'll set up a couple just little examples for you, because some of the stuff will be on microscopes, right? You need to be able to identify those white blood cells. You need to be able to identify some of the histology for the endocrine system. So those could be on there as well.
It'll be 100 questions, or are you doing?
It'll probably be 50 questions worth 2 is the typical there.
Okay.
Yeah. And that's another one. If you're like, how do I study the histology? You might look. I haven't looked around on the McGraw-Hill Connect if they have some good histology resources. I know people took pictures. You could turn those into little flashcards, or copy the pictures, out of your book for the different white blood cells, turn them into little flashcards, put the name on the back, something like that.
The library upstairs at CMC offers free color copies, which I thought was super helpful.
Yep.
For studying, because then you can mark things off and like hide the answers.
And I will say one of the things that's probably most useful to students usually is being able to study with someone, so like quizzing each other. And so you might set up a Zoom with each other, right? And like someone could share the screen and say, okay, what white blood cell is this? That sort of thing is often good for the person asking the questions and the person being quizzed. So yeah, feel free to use tech on that. But yep, it is memorization.
Yeah, can you hear me?
Yeah, Courtney, what's up?
I'm okay with memorization. I'm just feeling like I just don't know, like I can't seem to outline what I need to memorize because there's so much.
So
I haven't had a lab in 25 years.
Yeah. So those lists that I gave, right? So it's not like you're going to open the exercise and say, oh no, I need to know every single piece of the heart. It's just the stuff that's on that typed paper list. Yep.
So just look under the modules for your list.
Yep. Or if you picked up the paper handout in lab, they're the same, but the list is on the lab module as well.
Okay. I think I might. I don't know if I have all those. Maybe that's why.
Okay. Yeah. Look under the modules. If you're missing anything and can't find it, just shoot me an email.
Okay. Thank you.
Yep. You didn't have A&P I to make you memorize every feature of every bone. You're missing out. People don't usually freak out in A&P II because they're just like, hallelujah, this isn't near as bad as A&P I. I mean, it's like, whew, I got this. So.
Yeah, it's different online. I mean, we learned a lot though, a lot of systems and how things work. So it was a good base.
Yeah.
Yep. Yeah, but that definitely is the disadvantage to not having it in person.
It's just the practice on the memorization, but yeah. You got this.
Got it.
All right, duck out whenever you want. Otherwise, feel free to stick around if you want to keep chatting. But yeah, I'm done, so feel free.
Okay, Becky. So,
Carolina.
Okay, holy cow! This chapter, by the way, anatomy, easy breezy because of like anatomy 1. I was like, oh, it's like a lot, but it's fine because if I could memorize the bones, I could do this. Physiology part, still not loving it.
This is the hardest for sure.
The fluid, oh, hold on. It's like at the end of the chapter. Let me flip to it really quick. Oh, the fluid shift mechanism.
Okay.
So, in response to like high blood pressure,
yes,
that allows excess fluid to be pushed into the interstitial spaces in order to prevent the blood pressure from like going up. Is that correct?
Yes.
Yeah?
Yes. So
Okay.
tell me, so you're totally right. Let me see if I can do this. Okay, so when you're talking about fluid shift, in high blood pressure, what's changing about those 4 forces that we were talking about that would actually send fluids into that interstitial space?
Diameter? No.
No. So remember we had what? Blood colloid osmotic pressure, tissue colloid osmotic, blood hydrostatic, or tissue hydrostatic?
It would be just trying to put blood hydrostatic pressure.
You got it. Good. So all they're really saying in this idea of fluid shift is that if you have an increased blood pressure, right, and you're increasing that, basically that capillary hydrostatic pressure, or the blood hydrostatic pressure, we're pushing. And so all this water is going to start leaking out into the tissues. And that actually helps us lower blood pressure because we're decreasing the volume. So you're taking some of the pressure out of the tube by letting the fluid leak out.
Okay, so like in reverse, if it was starting to get too low, it would push some of that back out, which is the tissue hydrostatic pressure.
Yeah, so then if other systems maybe came into play and started lowering blood pressure, then all of a sudden, like, your body is like, whoa, why are all my tissues so full and swollen? And now that pressure would exceed the blood pressure, and you're totally right. Tissue hydrostatic pressure could push that fluid back into the capillaries, and that would raise blood pressure. Yeah.
Okay.
Good. You got
it. Okay.
You got it.
Thank you.
Yep.
All right. I'm going to get going then.
Sounds good.
That was my only thing.
Okay. Other questions?
Thank you. Bye, guys.
I actually had a question on the from the endocrine test.
Yeah.
Because I feel like I was kept hearing conflicting things about autocrine. Is it the cell that secretes it? If it's autocrine, is it only affecting the very cell that secretes it, or is it only affecting other cells of that same type?
Same type. So like the classic example is in lymphocytes. So like a helper T cell, when it sends out messages, they go to other lymphocytes. And I think they're still calling that autocrine.
Okay, I feel like a couple different sources, like.
Yeah, was there a specific question where that came up?
Yeah,